Hello, my name is Samuel Coleman, and I am a data analyst in the TAN lab at the H. Lee Moffitt Cancer Center and Research Institute. Today I am here to talk about our paper systematic evaluation of the predictive gene expression signatures of immune checkpoint inhibitors in metastatic melanoma, which was recently published in Molecular Carcinogenesis. First, an introduction. Immune checkpoint inhibitors, or ICIs, have changed the landscape of care in various cancers, including melanoma. The overall outcome of patients has improved due to the introduction of ICI treatment. However, this therapy only works on a subset of patients and is often associated with immune-related toxicities. In addition to the economic costs of ICI treatment, a need exists to develop a predictive biomarker which can help to select patients that will respond to ICI treatment. So we collected 28 previously published gene expression biomarkers of response to ICI treatment. In addition, 531 publicly available RNA sequencing samples from metastatic melanoma patients were collected. The RNA-seq data was log2 transformed to either transcripts per million plus 1, counts per million plus 1, or fragments per kilobase million plus 1, depending on the repository format. The gene expression across the samples for each signature was converted to a z-score before being averaged to assign a single score for each sample. This average score was then used to compare responders and non-responders using area under the receiver operating characteristics curve, AUROC, as the measure of predictive capability. Gene set enrichment analysis with the Hallmark gene set and TIMEX cell types was also performed to compare responders and non-responders. Finally, patients were classified as a responder or non-responder depending on their RECIST criteria. Complete and partial response were marked as responders, while stable and progressive disease patients were marked as non-responders. The results showed 18 gene signatures with an AUROC value of greater than 0.6. Additionally, 7 of these gene signatures returned an average value of 0.69 or greater. A Pearson correlation analysis was conducted using the AUROC scores to look for common themes among signatures. 2 clusters of gene signatures appeared, one of which were related to immune activation, which contained signatures like chemokines, interferon gamma, and MHC1 and 2. The other cluster contained immune suppressive related signatures like the Wnt and MYC signature. The AUROC performance was then stratified based on the time point at which the RNA-seq sample was collected. The RNA-seq data had 5 pre and 5 on treatment cohorts. We noted that the 18 gene expression signatures, which originally returned an AUROC value greater than 0.6, performed better in predicting responder and on-treatment samples in comparison to pre-treatment samples. This result may suggest that dynamic changes in the tumor immune microenvironment in patients after treatment initiation may assist in better predicting response to ICIs. A gene set enrichment analysis using the Hallmark gene set was also used to compare responders and non-responders. The responders at the top of these plots were enriched in pathways like interferon gamma, inflammation, and interferon alpha. However, the non-responders at the bottom of the plot were enriched in pathways like MYC, cell cycle regulation, and oxidative phosphorylation. The data was also interrogated using the TIMEX cell types. TIMEX is a novel deconvolution method for bulk transcriptomics based on single-cell RNA-seq signatures. TIMEX currently contains 37 cell type signatures, which are classified into 4 main cell groups: malignant, immune, stromal, and other, 14 major myeloid and lymphoid immune cell lineages, like conventional CD4 positive T cells, 15 minor immune cell lineages, as in naive CD4 and effector CD4, and 4 stromal cell types. Responders were enriched with infiltrating immune cell types like CD8 T cells, natural killer cells, and macrophages, while non-responders were enriched with stromal cells like fibroblasts and myofibroblasts. In summary, we gathered 28 previously published gene expression biomarkers predictive of immune checkpoint inhibitor treatment. These signatures had their predictive ability tested on 531 publicly available melanoma RNA-seq samples. Gene signatures related to immune activation, like interferon gamma signaling, chemokines, and immune cell infiltration and activation, have higher predictive power in this patient population. We also observed that on-treatment gene expression profiles have better predictive value, possibly due to capturing the dynamic changes in the tumor microenvironment induced by immune checkpoint inhibitors. Thank you.